and by now you must have gained clarity as to their deeper purpose in life rather than just pleasing our senses. Didn't you? Yes, right. Flowers are the centers of sexual reproduction in flowering plants, and we have already covered that in our previous modules and saw how skillfully each step leads to the next until we have a zygote. But what happens then? Well, after fertilization, a series of events occur in the zygote to develop into a seed and ovary into fruit. So, let's cover that in our current module. We shall examine post-fertilization events in flowering plants. These include development of an endosperm, development of an embryo, development of a seed, and formation of a fruit. Let's make a start with the first topic. Endosperm development precedes embryo development. We have seen how double fertilization takes place in our previous module. Once one of the sperms unite with the egg cell, a diploid zygote is formed inside the ovule, which lies inside the ovary. The other sperm cell merges with the polar nuclei to form the triploid primary endosperm cell. Hence, each ovule contains two fertilized cells, and this feature is unique to these flowering plants. So, what happens next? The nucleus of the primary endosperm cell now undergoes repeated mitosis division to form the endosperm tissue with no cell walls. But what is the role of this endosperm? Well, the cells of this tissue are filled with reserved food materials and serve as the embryo's food supply during the early development. So, do you like coconuts? Well, yes, who doesn't? The water in the coconut is nothing but the free nuclear endosperm, which is made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. However, in some plants like peas and beans, the developing embryo consumes the endosperm fully before the seed maturation, and in some cases, it may persist till seed maturation. For example, castor and coconut. So, here is an activity for you. Split open some beans, peas, groundnut, etc. and look for the endosperm. Do you find anything? Now let us move on to the next event, which is embryogeny, or the development of an embryo. This major event takes place inside the ovule of the carpel. But how? How does this happen? The diploid zygote divides many times by the process of mitosis to produce an embryo. The zygote divides to form a proembryo and subsequently to globular, heart-shaped and mature embryo. The embryo derives the nourishment from the surrounding endosperm and it continues to grow. It then differentiates to become a plumule, young shoot, radicle, a young root, and either one monocots or two dicots, cotyledons, which are the seed leaves. It is attached to the wall of the embryo sac by a suspensor. There are some characteristic features of a dicotyledonous and monocotyledonous embryo. In a dicotyledonous embryo, there is one embryonal axis and two cotyledons. The portion of the axis above the level of the cotyledons is the epicotyl, which ends at plumule or the stem tip, while the cylindrical portion Below the level of the cotyledons is hypocotyl that ends at the lower end in the radicle or the root tip. The root tip is covered with a root cap. In a monocotyledonous embryo, there is only one cotyledon, which is situated towards the lateral side of the embryo axis. Cotyledon is known as scutellum in the grass family. At the lower end, the embryonal axis has the radicle and root cap, which is enclosed in an undifferentiated sheath called the coleorhiza. Towards the upper side of the embryonal axis, epicotyls is present, which has a shoot apex and a new leaf primordial in a hollow four-layer structure, the cleoptil. Do you know most zygotes divide only after a certain amount of endosperm is formed? Well, yes. This is an adaptation to provide assured nutrition to the developing embryo. As the embryo and the cotyledons enlarge, they become crowded inside the developing seed and are forced to bend. Ultimately, the embryo and the cotyledons fill the seed, at which point the seed is ready for dispersal. You will be surprised to know that the embryo enters the state of dormancy or inactivity, and it resumes its growth only when the seed germinates. The developing seedling will rely on the food reserves stored in the cotyledons until the first set of leaves begin photosynthesis. Now, let's examine the development of seed and the formation of fruits. Immediately after fertilization, the zygote divides to form the embryo, while the ovule develops a tough coat called the seed. In angiosperms, the seed is the final product of sexual reproduction. A typical seed consists of a seed coat, cotyledon, and an embryo axis. Not only this, but the seeds can be divided into albuminous and non-albuminous, depending on the endosperm consumption during the embryo development. As the name indicates, the former retains the part of the endosperm. For example, wheat, maize, barley, castor, sunflower, etc. While the latter shares its endosperm completely during embryo development. For example, pea, groundnut, etc. Did you know that some seeds can remain in the state of dormancy for as long as 2000 years? Interesting, right? Yes, when the conditions are right, the seed will take up water through the micropyle by the process of imbibition. This triggers the beginning of the growth of the seed. So, can you think of any advantages of the seeds? Well, yes. They not only provide protection to the young embryo, but also being the product of sexual reproduction, they generate various genetic variations. Now, we are very well aware of the fact that seeds in angiosperms are covered. But what covers them is my question to you. Yes, it is the fruit. But how? How are fruits formed in angiosperms? Well, let's see. Right after fertilization, the flower loses its shine. The ovary ripens and the other parts of the flowers, like sepals and petals, wither. The ovary continues to grow until it develops into a fruit. The fruits may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut, mustard, etc. Now you must be thinking, what is the function of a fruit? Protection of seeds? Am I right? But if we eat them, how are seeds protected? Well, Think about it. Fruits aid in seed dispersal. Just like the flowers, they too have a bigger mission to accomplish, which is to land the seeds at proper conditions safely so that a new plant is formed. These fruits are not just walls for the seeds, but interestingly, they have walls too. And this is known as the pericarp. So, what exactly is this pericarp? Well, the part of the fruit formed from the ripened ovary is called the pericarp. Although in most of the species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. And such fruits are called, yes, such fruits are called parthenocarpic fruits. Can you name one such fruit? Yes, you guessed it right. It's banana. What about other fruits? Well, they grow from the ovary only, and so they are called as true fruits, such as guava, orange, etc. Some fruits like apples, cashew, and strawberry develop from the thalamus. Since they do not develop from the ovary, these fruits are called false fruits. And not just fruits, even seeds are formed without fertilization. And this is known as apomixis. Grasses show this phenomenon. Apomix, or a plant that reproduces by apomixis, have several advantages in horticulture and agriculture. Apomictic seeds can be formed in different ways. In some species, reduction division does not occur in a diploid cell, and embryo is formed without fertilization. In some other species, nucellus cells around the embryo sac divide and protrude into the embryo sac and develop into the embryos. So, here's an interesting fact. Humans have identical twins, and just like that, plants too show this alike feature in the name of polyembryony, PE. It can be defined as a simultaneous emergence of two or more seedlings from one germinated seed. Well, that's a lot of information about flowering plants. So now, why don't you just pause the video and recall the journey we've covered so far? Or let's do it together. Flowering plants or angiosperms are composed of calyx, sepals, corolla, petals, androecium, stamens, and gynoecium, the carpels. Pollen grains or the male gametes are created in the anther of the stamen. A carpel consists of stigma, style, and ovary. Ovary contains the ovule, where the female gametes or the egg cell is formed. Pollinators like bees or abiotic sources like wind and water aid in the process of pollination. Once the pollen grain has landed on the stigma, the pollen tube grows and the union of the sperm and egg cell takes place. Flowering plants exhibit a unique feature of double fertilization. The ovule develops into seed and the endosperm provides the nutrition to the embryo inside the seed. The wall of the ovary ripens to become a fruit, and other parts of the flower usually degenerate. And that's how we get a fruit. So, students, in our next module, we will discuss yet another interesting topic of biology. So, till then, why don't you explore some interesting creations of nature?